Okay, I'm all hooked up. Thank you. Thank you very much for the introduction. I thank the society and organizers for inviting me to Paris to present to the meeting today. So I'm going to talk about some of our work over the last quite some time related to obesity in children and the early life development, and hopefully there's some overlap in your interests as well. Our driving question, driving our research, there's two questions driving our research. One, the first question is, why is increased adiposity so bad for your health? What is it about excess adipose tissue buildup in the body that causes diabetes, cardiovascular disease, several forms of cancer, and so on? And second, when does that process begin? When does obesity start to turn bad? And a lot of that study is based on, historically has been based on adipose tissue location. This slide here just shows a couple of MRI images. We use fat imaging quite a lot in our research to locate where in the body fat is deposited. And you can see here two cross-sectional abdominal images from two different teenagers. They're both overweight, but you can see the fat pattern is very different. The red areas on those slices, this is a cross-section across the umbilicus. The red section is the fat, and you can see on the left, the image, there's a thick layer of adipose tissue around the periphery and several islands of fat within the abdominal cavity. That's visceral fat. Subject on the right, who has an equal degree of overall adiposity, has much less subcutaneous fat and much more visceral fat in and around the organs. You can see the large organ on the left is the liver. The liver on the left is brightly lit. That has a high excess of triglycerides in the liver. In fact, that child who came to us for a research study who was otherwise healthy ended up having fatty liver disease. You can also see the pancreas, which is the kind of long, thin organ indicated by the arrows on the left here. You can see the pancreas is literally, in this subject, swimming in fat. So the pancreas, which is hard to find the borders here you can see very clearly the pancreas surrounded by a lot of adipose tissue, triglycerides. A lot of our work over the years has like others, has shown that it's both liver fat and visceral fat that somewhat drives metabolic risk and obesity. I'm not going to talk too much about that today, actually. Another competing theory about why fat is bad for your health relates to adipose tissue inflammation. Just briefly as well on this topic, you can see histologically fat cells from this subject here. Both these fat cells were derived from, by biopsy from obese individuals, and you can see quite a striking difference in the adipose cells here and here. Here, the adipocytes from this subject are highly inflamed and surrounded by these what we call crown-like structures, which surround dead or dying adipocytes. And we published a paper a few years ago which showed that obese subjects with inflamed adipose tissues were the ones that had more metabolic, poor metabolic phenotype. So you can see here there's two groups of young adults. It's hard to do these studies in children because we took a fat biopsy. But you can see two groups of young adults who are equally obese. BMIs are above 35 on average, high percent body fat, and high amounts of subcutaneous abdominal fat. To look at them, they would be physically very similar, but we separated them into two groups. One, just purely based on the adipocytes having no inflammation, no crown-like structures in the fields of view, and those with numerous crown-like structures. And you can see that these two groups had very, very different metabolic profiles due to the association with adipose tissue inflammation. They're hyperinsulinemic, hyperglycemic, poor beta cell function, much more visceral fat, much more liver fat, and also had circulating levels of inflammation. And in relation to the previous talk, these subjects with adipose tissue inflammation had upregulation of the NF-kappa pathway. Now, of course, the main topic of what I want to talk to you about today is how this process begins very early in life. And of course, so obesity develops across the life spectrum. There are various critical periods for the development of obesity, most of which occur early in life due to pregnancy itself, the newborn period, adiposity rebound, puberty, and so on and so forth. These are all critical periods that have been identified that are critical for the development of obesity. And of course, pregnancy itself, maternal obesity is a critical risk factor, gestational weight gain, and gestational diabetes all affect obesity in utero due to fetal programming. This is one striking study. This is not our work. This is from Kevin Grove's laboratory at the Oregon Primate Center. And this is one of the most clear-cut examples of how fetal programming due to nutritional exposure can lead to obesity and a metabolic phenotype. And what's interesting about this study is that they show that the earliest sign of the fetal programming due to maternal high lipid consumption was fatty liver. So these were primates who were either control subjects or offspring from mothers who were fed a high-fat diet during pregnancy, some of whom were resistant, hence the R, to the high-fat diet. The mothers were resistant or sensitive, but in either case, the offspring of those animals, when they actually looked at fetal liver, it was much higher. Fetal liver was more than double in the offspring of the high-fat fed animals. That was one of the earliest metabolic indicators in this model of exposure to maternal high-fat diet. This is a study that was just published a few months ago. I'm sorry for the quality of this slide is not very good. But it's a human study in which they measured liver fat in newborns in the first, I think it was in the first week of life. It was just published in the Journal of Pediatrics. On the axis here is liver fat fraction by MRI, and on the x-axis is maternal pre-pregnancy BMI. And again, what's interesting is the unique metabolic difference that was different in the neonates was not total adiposity or visceral adiposity or any other metabolic parameter. It was actually liver fat in the first week of life was shown to be already significantly higher, shown by this correlation, either in mothers in the solid dots who were mothers with gestational diabetes or mothers with normal diabetes during pregnancy. Nonetheless, what the main point of this study is that the initial metabolic signal, or has been proposed as the first hit of maternal obesity on the newborn, is an elevation in newborn liver fat. So this is very relevant to our work for reasons I'll explain in the following slides. So our work is in children in Los Angeles, very high Hispanic population. This shows the prevalence of obesity at age 3 to 4 years of age in children in Los Angeles, going back to 2003 in various ethnic groups. And you can see the higher levels of obesity at age 3 is among Hispanics who are either of English-speaking mothers or Spanish-speaking mothers. And you can see this is continuing to increase, and the levels of obesity at age 4 are almost twice as high among Hispanic children compared to other ethnic groups. And fatty liver has been also been studied quite broadly in children. This is one study from Jeffrey Schwimmer's group in San Diego that actually documented fatty liver in children who died accidentally and livers were autopsied and analyzed for fat. And this study showed that many children had fatty liver, but you can see a striking ethnic disparity, which has been shown in other studies now since then. So African Americans just don't tend to accumulate liver in the fat in the liver. Whites, Asian, and highest among Hispanics. So the prevalence of fatty liver was almost 40% in obese children. And in fact, in our studies in Hispanic children, fatty liver is by far the most prevalent metabolic disorder that we have seen. Fatty liver is a spectrum of conditions, starting with hepatic steatosis, accumulation of fat in the liver, NASH, which is the inflammatory state, fibrosis, and then ultimately even in liver cancer. This is a spectrum, a continuum of conditions that begins with fat accumulation in the liver. Now, there's some very interesting genetic components to this, which also are quite specific to Hispanics. And some of this was derived from a GWAS in the Dallas Heart study, which identified this gene, which is a single nucleotide polymorphism. It's a C to G substitution. The function of this gene is not entirely known, but what's interesting is the effect of this gene is very high on fatty liver accumulation, and this gene is highly prevalent among Hispanics. So in fact, 50% of Hispanics in this study carry this C to G mutation, whereas only 10% of African Americans. Our studies published a few years ago verified the impact of this gene in children. You can see here Hispanics with the CC allele, the CG, and the GG. Almost 25% of the children were homozygous for this mutation, and as you can see, had more than a twofold higher fatty liver. Generally, the clinical cut point for fatty liver is about 5% fat in the liver. You can see African Americans were below that. No African American had the GG allele, and Hispanics with that were very high for fatty liver. And the effect of this gene we showed was manifested as early as 8 years of age. That's the earliest that we've studied it, but I'm sure in younger children this is probably true as well. To make this story more interesting, we also looked at the interaction of this gene with dietary components. We looked broadly across the diet to see which dietary factors might be related to fatty liver. No dietary factor other than dietary sugar came out of this analysis as being relevant, but dietary sugar, as you can see here, was not related to liver fat in those with the CC and the CG genotype, but in those in the very high risk group with the double G allele, you can see there's a positive correlation between fatty liver and sugar consumption, such that the combination of a high consumption of dietary sugar in children and the gene were associated with the highest levels of fatty liver. So here there's a gene by diet interaction between this PNPLA3 gene and dietary sugar. Now, another interesting finding that's actually relevant to some of the discussion at this meeting is something that we just stumbled across recently and is currently under review, is that the only other dietary measure in these children that we found to be associated with fatty liver was actually a protective role of vegetable consumption. And this is non-starchy vegetable consumption, particularly leafy green vegetables, which you can see are protective. So these are four groups. These are children in quartiles of vegetable consumption. And you can see here the lowest quartile with 0.1 servings per day, very small level of non-starchy vegetable consumption. And the fourth quartile is almost 2 servings per day. And you can see the highest quartile, and this is independent of those genes that I mentioned, as well as other characteristics such as dietary sugar. So independent of those factors, high levels of vegetable consumption are protective for fatty liver. And there's actually pretty good evidence from animal studies, practically none in humans, to show that high levels of polyphenols, and in particular catechins, are protective of fatty liver for a variety of different possible mechanisms, including decreased expression of lipogenic genes in liver, which would inhibit de novo lipogenesis, and an increased mobilization of liver fat through increases in beta oxidation. And it turns out that some of these compounds competitively inhibit the absorption of fructose and glucose transport in the gut, thereby limiting those monosaccharides, which are substrates, particularly fructose is a high level substrate for de novo lipogenesis. And there's also inhibition of intestinal sucrase by these compounds as well. So it may turn out to be a useful compound for limiting fatty liver. Now, sugar in early life. This is a study that we reported a few years ago that looked at the very early impact of dietary sugars on obesity in, this is again in Hispanic children who were aged 3 to 4 years of age. There's a lot of evidence in the literature now for dietary sodas and dietary sugars and obesity. This is some of the data amongst at the earliest ages of age 3, showing how this works. So these are children aged 2 to 4 years of age who were separated based on their level of consumption of sugar-sweetened beverages at age 2 to 4 years of age, 0 sugary beverages, 1 serving per day or greater than 1 serving per day. And you can see a very clear set of relationships here. We looked at this as a function also of breastfeeding. So among children who are never breastfed, you can see a very strong effect of consuming sugar-sweetened beverages at age 2 to 4 years of age. Almost 25% of these children are already obese, as defined by a body mass index above the 95th percentile. And you can see a very striking protection from obesity in those children who never consumed sugar-sweetened beverages. If you look at those relationships in children who were breastfed for 1 year or less, you can see a very similar, if not identical, pattern of relationship. There's still a very clear association with sugary beverage consumption, and there's no protection from breastfeeding regardless of sugar-sweetened beverage consumption. Interestingly, in those children who were breastfed for 1 year or more, and there was about 20% of these children who were breastfed for a year or more, you can see a very striking effect that, first of all, breastfeeding by itself is very protective in reducing obesity in all groups, and breastfeeding abolishes the negative impact of sugar-sweetened beverage consumption. So there's no relationship here in these children as a function of sugar-sweetened beverage consumption. So this raises the question as to whether dietary sugar is damaging in early life. And in fact, there's quite a bit of evidence to show that exposure of the developing fetus and the young infant to high levels of dietary sugar may be more problematic. And that's because in these periods, fat cells are developing, the body is obviously growing and laying down new tissue, and that's when these substrates have a high impact. So I don't have time to review all these today, but we just published this online in a review that I'll direct you to in Nature Reviews. It was just published actually last week, in which we reviewed studies showing that animal studies in whom the developing fetus or the newborn through breastfeeding were exposed to sugars early in life had a lifelong risk of obesity and metabolic risk. And this seems to be explained somewhat, at least by fructose, which affects fat cell and hypothalamic development. So exposure to fructose during pregnancy and during breastfeeding has multiple effects on adipogenesis. Fructose promotes adipogenesis. On hypothalamic development, fructose, for example, does not elicit a leptin response. So leptin is not in circulation and does not have its necessary effects on brain development, for example. This is a critical issue because fructose is increasing in consumption rapidly. Going back to the 70s and 80s, there's been a very rapid rise in fructose consumption due to the advent of high fructose corn syrup. And fructose, as a monosaccharide, has very different metabolic effects. Just very quickly, it's a much sweeter sugar. It does not elicit an insulin response. It does not elicit a leptin response. So it goes metabolically unchecked in the diet. Fructose is metabolized almost exclusively in the liver, where it is converted to triose phosphate and then triglycerides, packaged for either export into the circulation or accumulated in the liver, causing insulin resistance. So this is why high levels of fructose exposure are damaging to the liver. Also, this is a study published by my colleague earlier this year that used functional MRI to show that when young adults consumed fructose relative to glucose, the sugar affected different parts of the brain very differently. The orange spots are areas of the brain that responded more to fructose than glucose and included, for example, the hypothalamus. This is our study that actually analyzed popular beverages in the US that are made with high fructose corn syrup, which the beverage industry claims has similar or identical levels of fructose as if they were made with sucrose, when in fact we showed that Pepsi, Coca-Cola, and Sprite, these are the three most popular consumed beverages in the US, 65% of the sugars were fructose, not 55, which was the expected level if they were made with HFC 55, or 50% if they were made with sucrose. So you can see a much higher exposure to fructose, what we call hidden fructose, in the diet in these beverages made with high fructose corn syrup. So I think my time is rapidly coming up, so I'll just kind of briefly wind up here. Some people say, well, why is that a problem? If fructose is in apples and fruits and fruits, then why is fructose a problem? There's a couple of different answers to that question. The first is there's much less of it. So an average apple may have 10 to 15 grams of fructose, but that has other dietary benefits. And it's a question of the absorption into the system, because this fructose is released very slowly into the system because it's packaged inside the cell wall. There's no cell wall in sodas. It's just pure fructose, which is absorbed very rapidly and exposed in large amounts very quickly to the liver, rather than gradually over time. So just to briefly summarize some of the take-home messages from the talk today, thank you for your attention. Maternal diet, especially high fat, which has been shown in animal models quite clearly, and also emerging data from high sugars and especially fructose, contributes to fetal programming of obesity and metabolic risk. Liver fat accumulation appears to be a very early hit in the progression of obesity and metabolic risk, starting very early in life, if not during fetal development. Third issue is other factors contributing to fatty liver during life include genetics, ethnicity, and dietary sugar. There's some evidence, not in humans yet, but we need the data showing possible protective role of polyphenols from liver fat. Fifth, fructose is more obesogenic in multiple organs, including fat, liver, and the brain during critical periods of development. And this is exacerbated by what we call hidden fructose in the diet. For example, high fructose corn syrup, fruit juices. They're not made with added sugar, but contain high levels of natural fructose. Issues also affect developing fetus and the infant. Therefore, overall, the conclusion, we need a life course approach. We need to begin treatment in the developing fetus for lifelong protection and risk reduction from obesity and metabolic risk. Thank you very much for coming today. Appreciate it.
Thank you, Michael. This paper is open for discussion.
Thank you for a very interesting.
No.
Thank you for a very interesting talk.
Thank you. Now, my question is twofold. One is, are there fruits, because we're recommending 4 to 5 pieces of fruit a day, fruit or vegetables. So my question is, are there foods that are especially rich in fructose and that one should discourage people from having, even and more especially in natural juices? And the second is, these Hispanic children that you study, are they uniform? Whether they come from Peru or they come from Argentina, wherever, have you found them to be uniform?
Thank you. So in response to your first question, so the issue with fruits and vegetables in their natural whole form is really not an issue unless you're eating 5 apples at once, because the fructose levels are much lower, and as I mentioned, the rate of release in the digestive system is much slower.
No matter which fruit. Essentially.
Essentially, there may be some variations in fruit, maybe higher in fructose, but still you're not going to get the amount that you'll get in juices. Now, in an apple juice, which is, the juice from a glass of apple juice is maybe the juice from 4 or 5 apples, that's a lot of fructose. So in fact, I think probably apple juice in an infant who's developing may be at full strength may be just as bad as a soda in terms of the sugar dose. So in fact, we have a paper we're working on which we measured the fructose concentration in apple juice, and the sugar and the fructose composition in apple juice is higher than in some sodas.
And the children?
Excuse me?
And the Spanish?
And the Spanish children, yes. Thank you for that. We frequently get asked that question, and it's something we're addressing. I mean, there's evidence from other studies. We haven't looked specifically in all different countries, but what we're doing currently is we have this now in over about 400 or 500 children. We're measuring the genetic admixture that will be able to kind of quantify a bit more the ethnic racial issue to look at that question in particular. It's something we're very interested in, but there's several other studies from different countries that show this effect. I don't know if there's subtle differences within Hispanic or not. My guess is there's not.
Michael, how does orange juice compare, or some of the other common juices?
Well, I think they're just as high as fructose, just as high in fructose. I mean, if the natural fruit has fructose, then the
So
juice.
Juice would be as high as
Yeah.
apple juice?
Yes.
Thank you. There's a question up there, and then
All the way back at this corner, if I might.
Oh, hi, Steve.
Michael, the PNPLA2 is a very nice story. It's a phospholipase that encodes for a gene that cleaves phospholipid. It is one of a number of genes, and I think that it's important when you think about fatty liver that there's an input, fat, there's an output, the ability of the liver to secrete fat, and the net is what causes fatty liver. And so you might look at some of the genes affecting the output. So we have a paper earlier this year in FASEB Journal showing that we can predict quite nicely who is going to be susceptible to fatty liver associated with obesity in 845 people with biopsy-proven fatty liver. That if you look at the choline genes and the number of other genes that have SNPs in them, you can predict nicely who's going to be a poor secretor, and when they have high fat intake, they develop fatty liver. They don't if they don't have high fat intake. So you might want to expand your study to look not only at single SNPs, but patterns of SNPs that would affect fatty liver in the Hispanic population. It turns out PNPLA2 kicks out in our study as well.
Yeah, we haven't looked at. We've looked at a number of other SNPs, We have a paper coming out on that. GCK1 is also playing a small role, but we just looked at five genes. We haven't looked fully across the spectrum. That's a good idea.
And Billy?
Michael, I want to come back to the point of Hispanic, because I am Hispanic or not. I don't know what I am. No, really, it's genetically, I am 100% European. When I fill my papers in the United States, I put I am Hispanic, then he's Spanish, and then it's a big mixture. Probably, many of the people that you are studying came from Mexico, and they are closely related genetically to Asians than to Europeans, because the migration came from the north. How America was populated, it was from the north and with Asian people. Then probably they are genetically closer to Asian than to Europeans. Then I don't think that the main point is that perhaps for us as a scientist, we shouldn't use the term Hispanic because it's more American than worldwide.
Yeah, point. Yeah, fully agree. Most of the participants, I mean, I should say, more accurately, Mexican American, because 90% of the people that we study in Los Angeles are Mexican or Central American. And the admixture element is something we've been anxious to look at for a while, and we've just finally sent those samples off. We should have that data very soon, which will be able to quantify that much more specifically and scientifically.
Thank you very much. Unfortunately.